So I'd like to welcome you to the International Day of Women in Statistics and Data Science. This session is sponsored by Biomathematics and Statistics Scotland. I'd like to introduce you to Professor Rachel McRae, who holds a chair in statistics in the Department of Mathematics and Statistics at Lancaster University. She's the director of the National Centre for Statistical Ecology and a fellow of the Learned Society of Wales. Much of Rachel's research has been motivated by a desire to reliably inform conservation strategies, which often requires estimation of population sizes for species that are difficult to count. In particular, she's developed a number of new statistical approaches for the modelling of capture-recapture data, a topic which will be described in this presentation. Professor McRae's presentation is titled Statistical Models for Conservation Translocations. Thanks very much, Rachel.
Thanks very much, Esther, and also to Altea from BIOS, who has coordinated this session. I really do appreciate the invitation, and it really is a great privilege to be able to speak on such a special day. So the work that I'm going to be presenting today is very much not individual work, and given the focus of today's activities, I'd really like to draw attention to two female early career researchers who have really heavily contributed to this work. So the first is Katie Bickerton. So Katie was a PhD student with me at the University of Kent, jointly with the Zoological Society of London. And Katie's recently submitted her PhD, and a couple of the papers have been submitted which present the work in this talk that I'll give today. And the second is Faye Frost, who is a postdoctoral research associate with me, formerly in Kent and subsequently in Lancaster. And it really is important that their work is fully recognised, and really it should be them giving this presentation, because that they really have advanced the area. So as well as those two early career researchers, I mentioned that Katie's PhD was in collaboration with ZSL. It was also in collaboration with the Mauritian Wildlife Foundation. And you'll see that the applications within this presentation are motivated by case studies that we've been working on with them. So Katie was jointly supervised by John Ewen from ZSL and also Stefano Canessa from University of Bern. So during today's presentation, I'm going to give you a brief introduction to conservation translocations. I'm then going to present a new capture-recapture model. So this is a kind of modified version of something known as the Jolly-Sieber model. And this is specifically for translocated populations. And if you're not familiar with Jolly-Sieber models, please don't switch off straight away. I'm going to give a sort of an introduction to capture-recapture. So hopefully you'll be able to understand the gist of the model, even if you're not familiar with capture-recapture data. I'm then going to present two case studies, both motivated by work of the Mauritian Wildlife Foundation. And then I'm going to talk a little bit about some work in progress, because I always think you shouldn't just present work that's complete. It's really nice to see some of the challenges that we face during research. And I'll have, well, it's a fairly small slide of further work, but actually each of the points is going to take quite some time to deal with. So that's the kind of general structure for this afternoon. Okay, so first of all, what is a conservation translocation? So conservation translocation is when you deliberately move organisms from one site to another. And the reason why you might want to do this is because you want to see beneficial outcomes, either at population, species, or ecosystem level. So conservation translocations have been garnering pace recently because they're being increasingly used in programs to look at the conservation of threatened species, and also as part of, sort of ecological restoration programs, which is very much the motivation of the Mauritian Wildlife Foundation. So for those of you not familiar with the idea of conservation translocations, this is a really nice science article that came out earlier this year. And John Ewen, co-supervisor of Katie, is a co-author on this paper. And it really demonstrates quite how precarious the species are on our planet at the moment. So there are actually many species which can be considered to be extinct in the wild. So part of the conservation translocation strategy is to think about, could these species be taken from zoos and translocated back into form wild, self-sustaining populations? So I got involved with this work through an EPSRC proposal, and we had a number of aims of the project. And one of them was to think about, well, how do we determine whether a translocation has been successful or not? So obviously one of the main aims of a translocation is to determine whether the individuals who have been released into an area, whether they've survived adequately and whether they've produced enough offspring to be able to establish a new population. So there's no point translocating species back onto an island population where they've subsequently been before, if it's highly likely that those individuals are just going to die out again. But it can be really difficult to determine whether a translocation has been successful. So obviously post-release of a translocation requires intensive post-release monitoring. And in particular, the species we're going to be dealing with, they're going to be really difficult to detect. So capture probabilities of individuals during that post-release monitoring phase are going to be very low. But even despite that sort of low capture probability that we're going to be faced with, we still need to be able to come up with methods to be able to robustly estimate population size. And crucially, it's really during that early establishment phase of the population where it's going to be most valuable to get reliable population size estimates. We've also, on the other hand, got to think about, well, where are these species coming from? So I mentioned for the science paper that sometimes these are taken from captive populations, specifically bred for the purpose of reintroduction, but sometimes they can also come from relatively small source populations. And that's the case with the translocations that we're going to be looking at today. So we're taking individuals from a typically small source population. So we need to be really careful that we're not going to adversely affect the source population. So once again, to be able to determine this, we need to be able to obtain robust estimates of population size of the source population, both before we actually remove individuals and also following removal of individuals. So for both of these challenges, both on to determine whether we've had a translocation success or to think about the impact on the source population, the motivating desire is that we need to be able to sensibly estimate population size. Okay, so the type of data that we're primarily going to be working with during this presentation is capture-recapture data. So I apologize to those of you who are completely familiar with capture-recapture data, but because it may have been quite a broad audience, I just want to go through the structure of the data. So capture-recapture data relies on individuals being uniquely identified. So sometimes individuals might have a ring attached to them, for example. So you can see on this picture of the cormorant, it's got a green ring around its leg, and that ring will have an alphanumeric code so that when that cormorant is observed through binoculars or captured in person, you'll be able to determine which individual that is. So because we have that individual level of information, once that individual has been marked, it's released back into the population, and subsequent attempts are made to capture that individual. And the data that we'd collect for that individual is then in the form of a vector of zeros and ones, where a 1 indicates capture and a 0 indicates non-capture. So the first one of the individual typically indicates when the individual will have had a ring put on it, so when it will have been originally marked. So the other pictures on this slide, you can see some silver rings here. So this is an example of some historic British Trust for Ornithology rings. So you can see that there's different rings for different sizes of birds. And the rings on the far right picture are the rings that are currently used on the Isle of May seabird colonies. So you can see that over time that the material with which the rings are made from, these blue, yellow and red rings are very lightweight, so they're much less likely to adversely affect the birds which they're put on. Now, sometimes, depending on the species that you're dealing with, a species might have its own unique markings which allow it to be identifiable. So you can think of this as like a fingerprint in a human. So the species that's shown here is a great crested newt, and great crested newts are identified because they have this striking yellow and black belly pattern. And that yellow and black belly pattern is like a fingerprint for that newt. So that the layout of the black and the yellow marks will uniquely determine which individual that is. And the two study species we'll go on to look at later, one of them has its own unique markings, whereas the other relies on artificial tagging. So the type of data you have, you can think of this, if you've got an encounter history, so this vector of information of zeros and ones for each individual that you've captured in your population, and you put all of those row vectors together, we essentially have a matrix of zeros and ones. Okay, so the Jolly-Sieber model is one of the most basic capture-recapture models that allows for the population to be open. So it allows for individuals to arrive and depart while still being able to estimate population size. So that's really the structure of the model that we need here. So we know that the population is going to be open. We know that certain individuals are going to die during the period of the study. But really our parameter of interest is the population size. So just introducing a little bit of notation, suppose that HI is the encounter history of individual I. So you can think of that as being the row vector for individual I. And we calculate some sufficient statistics from that encounter history. So we can say that suppose individual I is first captured on occasion FI. So that will denote the first one that appears in that row vector. And suppose it's last captured on occasion delta I. So that will be the last one in the row vector for individual I. And we've already commented that the value within that matrix will be 1 if an individual is captured and 0 if it's not captured. So you can see here, we're just using this notation X to denote that whole matrix of data. So you can think of the HIs, if you put all of those row vectors together, you'd form matrix X. And we can also think about, well, what's the size of that matrix? So the number of observed individuals will be the number of rows in our matrix. And we might want to impose some sort of constraint on individuals that that new arrivals can only start entering a population at a given time. And that will be particularly important for the translocation work that we're doing. So I'm just introducing a parameter kappa here. That means we're constraining that individuals cannot arrive before time kappa. Okay, so the parameters of the Jolly-Sieber model, there are 4 parameters. So we've got a survival probability, which we allow to be time dependent, which we denote by phi of t. So phi of t is the probability an individual who is alive at occasion t remains alive in the study area until occasion t plus 1. And we've got the capture probability. So obviously we've got imperfect detection here. So we have to account for that imperfect detection via the parameter pt. And the beta t's are going to correspond to the proportion of individuals that arrive between occasion t minus 1 and occasion t. So we express this in terms of a proportion because we ensure that we cannot have more individuals leaving the population or more individuals so that our population size becomes negative. So you can think of these betas as probabilities. So what's the probability of an arrival between certain time points? And then finally, we've got our population size, which we denote by capital N. But in fact, so capital N will denote what we refer to as the superpopulation. So capital N is the total number of individuals who have ever been available for capture during a study. But really what we're interested in is to think about, well, what's the population size at a given time? And in fact, we can calculate that through some recursions in terms of the parameters as presented here on the slide. Okay, so the likelihood for the Jolly-Sieber model, it might look slightly horrendous, but I promise you it's not. So this is a multinomial likelihood. So we've got a multinomial coefficient here, and you'll see that we've just dropped a d factorial, which is why we've got a proportion here instead of equality. And the P of H0 corresponds to the probability of the 0 encounter histories. So essentially, we're going to have observed D individuals, so we can calculate the probability of each of those D individuals, which is given by the probability of HI. But there's going to be a certain number of individuals, N minus D individuals, which correspond to the number of individuals that we've never observed. And so the probability of never observing an individual is a really important part of being able to estimate population size. And I'm not going to dwell on the actual structure of these probability of observed encounter histories and probability of unobserved encounter histories, but essentially there are different components here. So you can see that the betas model the arrival, the phis model the survival from the time at which they're known to have arrived to the time at which they're known to have left. And then there's a departure probability, which is your 1 minus phi. And then this last term in the curly brackets corresponds to those captures and non-captures during the time in which the individual is available for capture. And you can see that the equation for the probability of observed encounter history for individual I looks very similar in structure to the probability of never being encountered. But the main difference is that previously we recorded the information of first capture for individual I, which is denoted by Fi. So the summation for possible arrival times only goes up to that point. And we denoted the last observation time as delta I. So that's the first time at which the individual might have left the population. So we allow the possible departures to start from that point. With unobserved individuals, we don't know those times. And so you can see we sum over all possible arrival and departure times for those unobserved individuals. Okay, so one of the main problems that Katie faced when she was starting to think about, well, how is she going to estimate, or how is she going to try and estimate population size following a translocation of a species, was that her default was going to be, well, I'm going to use a Jolly-Sieber model. But what we actually found was that if you try and use a standard Jolly-Sieber model, and it's very difficult to try and get the parameters of interest to be estimated from that model. And the main reason is that more often than not now, people are using R packages to be able to fit these capture-recapture models, but it wasn't possible within those R packages to specifically constrain the parameters that needed to be constrained. And so we thought, actually, we might want to slightly differ the structure of this model, and we might want to account for the translocated individuals differently to those wild-born individuals that will be the progeny of the translocated individuals. So the main issue is that if you fit a standard Jolly-Sieber model, you tend to, with the Jolly-Sieber model, you tend to not know the population size at the start of the study. Whereas obviously with a translocated population, we know exactly what the population size is, but it's very difficult to constrain the standard Jolly-Sieber model to account for that known population size. So what we decide to do instead is that we form a separate probability of observed encounter histories for the translocated individuals. And what we're actually doing there, we're conditioning on that first release within the population. So we're not trying to estimate population size at that point because we know what population size is. But conditioning on that release, we can then go on to calculate the probability of the remaining observed encounter histories for those individuals. And the structure of that probability of the translocated individuals is given on the slide. So what we have for this adapted translocation capture-recapture model is we still have the n factorial over n minus d factorial. We still have the probability of unobserved individuals raised to the power n minus d. And we still have these, the probability of the observed encounter histories for the wild-born individuals. So these probability of HIs now correspond to wild-born individuals. And what we've now got, we've got this separate term which correspond to the probability of the observed individuals for translocated individuals. So the notation here is that we're assuming there are N0 translocated individuals. And so D minus N0, so D is the total number of individuals we've got encounter histories for. D minus N0 of those will be wild-born individuals. So by partitioning the likelihood in this way, we're able to better control, we're able to say we know the population size at the time of the translocation. And really, we're using the Jolly-Sieber element of this model to be able to model the wild-born individuals, but we're using information from those translocated individuals. Okay, so that's all the kind of boring equations, let's actually think about how this works in practice. So the first case study that I'm going to talk about today is of a species called the lesser night gecko, which is a tiny gecko species, which is endemic to the Mauritian islets. So this picture, it might not be very clear on the screen, but this is somebody's fingertip. And you can see that this gecko kind of happily sits on somebody's fingertip. So lesser night gecko are considered vulnerable. There are very few wild populations left in the world. And Nick Cole, who was the supervisor of Katie at the Mauritian Wildlife Foundation, and Durrell, set up this plan because the aim was to be able to establish a new population of lesser night gecko on Round Island. So here we've got Gunner's Coin, which is an island off the coast of Mauritius, and you can probably tell from the shape of the island on the picture, but this island on the bottom is Round Island. So for those of you not familiar with the layout of Mauritius, so you've got main island of Mauritius, and then off the coast of Mauritius, you've got a number of small islands. So you can see that Round Island here is off the northeast, and then you've got tiny rocky islands of Gunner's Coin and Pigeon House Rock just to the west of it. And then in the southeast, which I'll come on to later, we've got a number of small islets. Now, Katie's PhD was badly hit by the COVID pandemic. So the primary aim of her project had been that she would be desk-based for the first 6 months, and then she'd be going out to Mauritius and actually carrying out the translocation of these geckos, and then having a couple of years of post-translocation monitoring. Unfortunately, that coincided with COVID. And so she wasn't able to do the translocation particularly early during her PhD. And so we were very fortunate that there had been a previous translocation of lesser night gecko, and there was really good data that had been collected from post-release monitoring of that population. So back in 2011, there had been a translocation of 30 gecko from Île Vacqua, this lower island here, onto Île Marianne. And so we could test some of our ideas around how we might model the capture-recapture data for the translocation that Katie subsequently went on to carry out, but we're able to test those ideas on that historical data set. Okay, so I'm not going to go into sort of huge details. Obviously, there's been an extensive amount of model selection and deciding which parameters needed to depend on which covariates. But one aspect that I wanted to show you was how we're able to estimate population size and how it differs to the best we could do with the existing Jolly-Sieber model. So what's shown in this plot, so this is the analysis of the historic lesser night gecko translocation from 2011. And you can see that there's been data collected, it's still collected now. So really good, consistent amount of data that was collected. And the darker of these lines are the population estimates that we obtain using our new capture-recapture model for translocated individuals. What's shown in the lighter grey were the estimates for population size based on trying to constrain the Jolly-Sieber model. And you can see that there really is quite a substantial difference between the dynamics that are estimated from the existing Jolly-Sieber model compared to the translocation model. But importantly, the aspect that's most important for determining whether there's been a translocation success is in these early phases of the post-translocation monitoring. So you can see that the dotted line here corresponds to the number of individuals who were translocated. And you can see that the Jolly-Sieber model estimated that these dipped quite substantially and actually got very close to zero in the later part of 2011. Whereas the estimates from our modelling approach shows that there was, of course, a slight dip. There's always going to be imperfect survival in those first months following translocation, but you can see it doesn't dip as far as the estimates that you would have obtained from the Jolly-Sieber approach. So in particular for this type of analyses, which really relies heavily on obtaining robust estimates of population size in those early phases, it's looking like our approach is a really important contribution. Okay, and as if the COVID pandemic wasn't enough for Katie to deal with, in the summer of 2020, there was a massive oil spill off the coast of Mauritius. And at that time, it wasn't really, known how this oil spill might affect the lizards within the island. So as well as sort of adapting, I'm going to be using historical translocation data for part of her thesis. She was now tasked with, can we use some of the capture-recapture data which has been strategically collected on these islands and consistently collected for a number of years to be able to work out, has there been an adverse effect on those populations? And obviously, in particular, she was interested in the impact on the lesser night gecko. So there were 2 islands of data where there was the lesser night gecko population. So if you recall that the source of the lesser night gecko for the translocation was from an island called Île aux Vaques, and they were moved on to the island called Île Marianne. So what we've got on the right-hand side are estimates from capture-recapture data from Île aux Vaques, the source population, and on the left we've got data from Île Marianne from the translocated population. So it looks like we're missing data here. So obviously we know that there were lesser night gecko on Île aux Vaques from early on, but previously distance sampling had been used to monitor the species, and capture-recapture data was only collected from 2016 onwards. So that's why we've got estimates, population size estimates from Île aux Vaques from 2016 onwards. So these population size estimates from Île Marianne are the same as the ones on the previous slide, but now we've got additional data. And the red dotted line corresponds to where there was the oil spill. So a couple of points to note here. So the first is that it doesn't look like we have enough evidence to say that there's been an adverse effect on the population size for either of these species, but it could just be that we're not able to detect an effect at this stage because of the amount of data that we've got. There is also a very large gap between data that was collected pre-COVID and when they were allowed to start resampling and surveying the areas again. So you can see that in the early years, there was a lot more monitoring that was carried out on these islands, but it was before 2020 that we had a data point here. And in fact, the subsequent data collection wasn't until quite a way through 2021. So we don't have data that's been collected kind of immediately post oil spill. And similarly from Île aux Vaques, you can see Île aux Vaques is a much smaller population, as evidenced by this sort of much greater uncertainty in the confidence bands that we've got. But again, there doesn't seem to be an obvious effect of the oil spill. So there could also be some confounding factors here. So obviously without humans visiting these islands during the COVID pandemic, that might have actually been very positive in terms of the establishment of populations. So it could be that that helped counteract any effects of the oil spill as well. Okay, and then to finish on a brighter note of Katie's thesis. So at the end of last year, she did finally get to do the translocation of the lesser night gecko that had been planned. And the post-release monitoring has only been happening in the last few months. But at this time, we haven't got any new individuals that are being born into the population because we wouldn't yet expect that. So it's actually with the lesser night gecko, you tend to only catch the adults rather than juveniles. And so we wouldn't expect to be able to start to detect new adults until next year. But what we are able to estimate are the survival probabilities that we're from the capture-recapture data that's already been collected on the translocated individuals. So as part of the release strategy for this translocation, there is some sort of expert solicitation in terms of how you should protect these lesser night geckos. So there were 4 different release sites, each with certain properties, and they were fenced and enclosed for a period of time before the enclosures were taken down. So you can see that we do have differences in survival probability between the 4 release sites. And sorry, I should have labelled that the red estimates here are female survival and the blue estimates are male survival. But these survival probabilities are well within the bounds that we'd expect the population to be able to establish itself, so long as the reproductive effort is sufficiently good. Okay, so that's case study 1. So that was it was a roller coaster of a PhD project, and Katie did a fantastic job of navigating all those challenges and really coming up with some important contributions. So the second case study is still again from the Mauritius Wildlife Foundation, and this time it's on a population of telfair skinks. So telfair skinks are at the other size dimension compared to the lesser night gecko. So they're the largest living species of Mauritian skink. And basically the telfair skink has declined since the 16th century. And it's believed that the decline of the species was really aggravated by the invasive species that had come onto the island, in particular the rat population that's come onto those islands. Now, in fact, the telfair skink became extinct in most of the Mauritius archipelago by around the mid-1800s. And there was one exception to that. So the exception was Round Island that we've already mentioned. So again, this is a historic translocation that's happened, but there was a lot of conservation effort to try and remove those invasive species. And so rats and rabbits were eradicated on Gunner's Coin between 1995 and 1998. So we've got Round Island where we've had the telfair surviving, and now they were trying to make Gunner's Coin habitats suitable for reintroduction of telfair skinks. So there was an ecological assessment of Gunner's Coin, and it was found that it would be suited to the reintroduction of telfairs. And 250 telfairs were translocated from Round Island to Gunner's Coin in 2007. Now, following on from that, 100 further skinks were reintroduced in 2012. And this is quite often a strategy that you have a 2-stage reintroduction. It's not an aspect I know a lot about, but it's to do with the introduction of genetic diversity into the population. Now, I should have mentioned earlier, the lesser night gecko, they have unique markings, and you're able to take photographs of the lesser night gecko and use clever computer software to be able to determine which geckos are the same one from the photos. With the telfair skinks, that's not possible, but all released and newly captured skinks, they were fitted with a microchip, and it was that microchip that contained that individual level information for capture-recapture data to be collected. There was also a point during the establishment of the telfair skink population in the reintroduced population where some individuals were removed for further conservation translocation efforts to those southeast islands that we saw on the map earlier. And we'll see those removals later in the study, and in fact that motivates the work, the second part of this talk. So we used, again, the capture-recapture translocation model, so the adapted Jolly-Sieber model, to be able to fit the model to these data, to be able to estimate the parameters of interest. And this work was carried out by Faye Frost, who was a postdoc with me on this EPSRC project, and she did a fantastic job of performing really rigorous model selection and exploring potential covariate structures which would be useful to understand the dynamics of the telfair skinks. So we've got 2 different plots here. So the first are the estimated population sizes for the estimated male population. So the green dotted lines here correspond to the points at which the translocations happened. So obviously we had the initial translocation, and then I mentioned there was that subsequent translocation in order to introduce some genetic diversity. So those are both denoted by green. And then the red dotted lines correspond to some times where some removals occurred for subsequent translocations. So we don't have capture-recapture data collected at those same time points, and unfortunately, we don't have the information about which individuals were removed from the population, and I'll come on to that later on. So you can see that there seems to be a nice increase in the population size, and it's fairly convincing that this population has really established itself now. And the dynamics look very similar for the female populations. And so this new population of tailfair skinks is going to be a valuable tool for reintroductions elsewhere. Okay, so in the last part of the talk, I want to think about slightly more about this source population and how do we actually model the effect of the conservation translocations on that source population. So as I mentioned, we are often taking individuals from very small source populations. So it's really important that we understand the effects of those. Now, removal and depletion models have been used to model removal of species prior to land development, and have also been used a lot for modeling eradication of invasive species. So the basic removal model is a very simple geometric model. So you can think that it's very similar to capture-recapture data, but you only ever capture an individual once, because when you capture that individual, you then remove it from the population. So thinking about how to construct the probability that you capture an individual at time t, you must have failed to capture it t-1 times and then captured it on the tth occasion. That geometric model assumes that the population is closed, which obviously isn't always the case. And this geometric model has been extended in a number of different ways. So, for example, my colleague and I, back in Eleni Matahu, back in 2016, extended that model to allow for the populations to be open, so to allow for new arrivals and departures during the removal study. And our PhD student, Ming Zhu, developed a removal model which accounts for temporary emigration of individuals. So allows individuals, it was actually for a lizard species that buries under the ground and then is only available for capture when it's above ground. Okay, so what do we actually want to be able to get from removal data? So we want to be able to assess the impact on the source population. So we want robust estimates of NT during the time in which removals are occurring. So the actual removal data, even though I said, well, it's very similar to capture-recapture data, but there's only a single capture for any individual, you can compress removal data to just be counts of the number of individuals removed at time t. So quite often you'll see removal data just reported as a vector of counts. And when you've got a very small number of sampling occasions, it can be really challenging to estimate the size of the population at time t when you've only got removal data. And that was really the sort of motivation behind the work that we've done. So what we wanted to think about was, is there any way we can supplement removal data with another form of data which will allow us to better estimate the parameter of interest, so time-dependent population size. So given my sort of history with capture-recapture data, the most obvious thing is, well, could we supplement the removal data with capture-recapture data? So what I'm presenting here in this diagram is we're thinking about if we had a period of time, which I'm denoting TOR, in which we perform capture-recapture on individuals. And then after time TOR, we start removing the individuals. So this would be a little bit like you might spend 2 or 3 sampling occasions where you go and capture the individuals. And even though this is subsequently going to be a removal experiment, you re-release those individuals. So this might take a bit of convincing of the people who have to do the field work, because they put a lot of effort in. It can be really hard to capture these individuals. So this is the structure that we've got. So N is our population size of interest. So the dark red corresponds to individuals that we observe during that capture-recapture phase of the experiment. And the pale red corresponds to individuals that we fail to detect during that capture-recapture period. We've then also got this dark blue period. So dark blue corresponds to individuals that we've removed, and light blue corresponds to individuals that we haven't removed. And so you can see that each of the capture, recapture and the removal parts have two possible outcomes, so observed or unobserved. And then we've got four possible combinations of dark red, dark blue, dark red, light blue, and so on. So that's the sort of general structure that we're thinking about. And I won't go into the detail because of the available time, but we can construct this, a likelihood for this model in a very simple way. It's very similar in structure to the likelihoods that I've expressed earlier. So we're going to have a form of capture probability. So that's going to be time varying. So that capture probability is going to be assumed to be the probability of capturing up until time t, tau, and the probability of capturing and removing for times tau plus 1 up to the end of the study t. And the likelihood of the joint data is given by this expression. So I'm not expecting you to absorb all the equations here, but really I've put it onto the slide to demonstrate that the structure of this capture-recapture model is very similar to the capture-recapture model that I showed earlier. And there's a few technicalities about the last capture of an individual and so on. So the model that I'm presenting here is now a closed population model. So this was our starting point, that we're not allowing the population to be open. So all individuals are available for capture during the whole of the study period. Okay, so what we want to think about is, suppose we've got a set number of capture occasions, or a set number of sampling occasions, I should say. How many occasions of capture-recapture do we need to be able to aid that estimation of n? So what's on the x-axis here are the number of occasions of capture-recapture. So in total, this is only a 6 occasion study. We've got a low detection probability of 0.04, and our true population size is 200. And as I mentioned at the start, when you've got low capture probability and a small number of occasions, it's very difficult to robustly and accurately estimate population size from removal data alone. And that's shown in these first pale purple box plots here. So what we've got in the left plot is the maximum likelihood estimate of total population size n, and what we've got in the right plot are the maximum likelihood estimates of the capture probability. So you can see from removal data alone, we're overestimating detection probability, and that overestimation of detection probability results in an underestimate of population size. Now, even when you supplement that removal data by allowing one of the sampling occasions to be capture-recapture instead of removal, you can see that we do improve the estimate of population size. And I'm not going to say that these are perfect estimates of population size, because that there is a little bit of negative bias here, but we're doing a lot better at trying to estimate that population size relative to having no capture-recapture data at all. And really, we were quite surprised that even increasing the number of capture-recapture occasions all the way up until 5 here, so that's assuming that we only perform removal on the last time, you can see that the estimate of n doesn't vastly improve. So we are in a situation here where p is very small and the population size is relatively large, and importantly, the total number of sampling occasions is fairly small. But you can see that even a small amount of supplementing the removal data with capture-recapture data has been really valuable. Okay, so that would be great if our population was closed, and also if we're in a situation where we know which individuals correspond to which part of the data set. So in the previous diagram, these two parts of the matrix were joined together. So that assumed that we know which individuals contribute to the capture-recapture data, and then we know whether those individuals were subsequently removed. In reality, these types of experiments are really conducted in a kind of disjoint way from one another. And so when individuals are removed, they don't necessarily record which individuals have been removed. And this is exemplified by the Telfair skink analysis that I showed you earlier, where we had those red dotted lines that correspond to removals of individuals. We know the number of individuals that were removed, but we don't know which individuals were removed. And so this structure is much more likely to be what we end up having rather than that perfect structure that we had before. Now, the main problem when you disconnect these two types of data, we don't know capital D. So if you remember, capital D denotes the number of individuals observed during the study. So if these are disjoint, we'd know little d, so we'd know the number of individuals observed during the capture-recapture part of the study, and we'd know what we're denoting by d star, which is the number of individuals observed during the removal part of the study. But we haven't directly got an idea of what capital D is, because we don't know what the overlap is between the individuals who are observed in the capture-recapture and the individuals who are observed in the removal. Now, for those of you who are familiar with integrated population models, it's quite common when modelling different types of data to say, okay, well, can we assume that these two data sets are independent, and can we construct a likelihood for the capture-recapture data set, likelihood for the removal data set, and form a joint likelihood by multiplying those two likelihoods together? And that was our default. We wanted to assess, well, how sensible is that approach, and how much information might we be losing? So what we've done here, we've set up the data, so we're partitioning the data into a capture-recapture component that we're denoting by Z and a removal component that we're denoting by Y. And then we're able to construct a likelihood for the capture-recapture component, a likelihood for the removal component. And then we can assume independence, as I said, and we're going to form what I'm referring to as an integrated likelihood as the product of two likelihoods. Now, that seems like a very sort of simple approach, and we really wanted to assess, well, how sensible is this? So algebraically, we looked at, well, what is the structure of the true likelihood? So if we did know the individuals and which ones contribute to which component data set, and this approximation where we're just assuming everything is independent. And really interestingly, after many pages of paper, because I haven't done so much algebra for quite some time, but we were able to show that the only way that that integrated, like, approximate approach and the true likelihood where we know the individuals differs is through the multinomial coefficient term. So the integrated likelihood has two multinomial coefficients. So the first is the multinomial coefficient that corresponds to the removal data. So the D star is the number of individuals observed in the removal. And the D here is the number of individuals observed in the capture-recapture. And then I'm denoting the rest of the likelihood function by capital lambda. If we compare that to the true likelihood where we know the individuals, what we find is that we've got a single multinomial coefficient, which is in terms of that capital D. And then that's multiplied by the capital lambda term. So, as I said, when we don't know the individuals or which individuals correspond to which data set, we don't know this capital D. But what we thought was, well, is there a way of approximating or estimating this D to enable us to essentially come up with a better approximation to the likelihood that more closely resembles this lower true one, rather than naively having these two multinomial coefficients? So that's what we're trying to do. So we're trying to come up with this approximate likelihood. And instead of having D here, we've denoted this by D tilde. So we want to come up with D tilde, which is an estimate of the number of observed individuals, taking into account the capture-recapture and the removal data that we've got. And we were very excited by this because, in essence, it seemed to us logical that we should be able to estimate D, because it's essentially the number of individuals that we've observed. And so initially we thought, okay, well, can we directly estimate D tilde? Can we introduce a new parameter into the model and estimate D tilde? And our numerical optimization was failing. It was always putting it on a boundary. So we wondered whether it's just an optimization problem. Then we thought, okay, we'll use an EM algorithm because that nicely suits that we want to estimate and then we want to maximize. And again, we ended up at the same point as the numerical optimization did. So essentially, we're in a situation where we can't estimate that parameter based on the information that we've got because we don't know those individuals. So that was a real shame. That was a real sort of gutting blow when we really thought that we'd done all the hard algebra. We've proved this, and so surely now being able to estimate it should be fine. So what we've ended up doing instead is, for those of you familiar with estimating population size, we've taken a Horvitz-Thompson-like approach. So essentially, we estimate all of our other model parameters, so the capture probability in this case, and then we use an approach to estimate n based on an estimate for d tilde that we have through evaluation of the MLEs from the capture-recapture component. So it's not yet complete. So this is very much work in progress. So if anyone has any really cool ideas on how we can tackle this, we'd love to hear from you, because I feel there should be a fourth bullet point of this is how we're really going to do it, because the third one just seems a little bit unsatisfying given we've got this relationship between the two likelihoods. So I just wanted to show you some simulation results from the approaches. So just to explain what we've got here. So these are box plots over multiple simulations of the estimate of population size. So these are the estimates from capture-recapture data alone. So you can see that we're underestimating population size. Removal alone, we're underestimating population size. Here is where we assume that we know the overlap between individuals between the capture-recapture and removal. So you can see that we nicely estimate population size here. The orange plot is the naive integrated approach. So where we simply multiply the two likelihoods together, we don't worry about the fact we've got two multinomial terms. And then what we've got is using our Horvitz-Thompson approach, this pale brown plot. So you can see there's a kind of slight improvement compared to the naive integrated approach, but it's certainly not as good as knowing the overlap of individuals. So I'm hoping in future work we'll have a nice plot somewhere between the pink one and the brown one once we've figured out the next best thing to do. Okay, so I've mentioned this is very much work in progress. So if anyone has any ideas, please feel free to contact me either through the chat now or by email subsequently. I really love to develop new collaborations with people. What I've presented for the joint capture-recapture and removal work, that's currently for closed populations. We have extended it for open populations, but I didn't have time to present that today. We want to think about how we can take our approach and try and think about that Telfair case study where they had those removals that occurred partway between the capture-recapture sampling occasions, and whether there's something that we can use our new structure, our new model for that to better estimate population size at those times. And really thinking about how can this be used as a tool for future conservation translocations, is that we'd like to think about sensible ways of forecasting the effects of removals from source populations so that it can very much be used as a planning tool for subsequent conservation translocations. So there's a few references there. I've mentioned both of Katie's papers. So one is now in press and the other one's in preparation. So the one in preparation is on the oil spill work. We haven't yet written up the Telfair analysis, but hope to soon. And there's a couple of papers there that we present those extensions to the removal models. So finally, I'd just like to say thank you. So this work has been a sort of huge collaborative effort. As I've mentioned, there's lots of people that have been involved with it, and also lots of people who have provided funding for the work as well. But I will conclude there. And thank you very much for listening and attending the event. I really appreciate it.
Thanks so much, Rachel. That was absolutely brilliant. If anyone's got any questions, can you please type them in the chat and I can pick them up. So while still waiting for that, I have one. I just wondered whether the skink or the geckos actually move between islands, either sort of transported by humans sort of accidentally, or whether they swim, perhaps.
I'm not aware of them swimming. I have to say I'm not sort of hugely comfortable with the ecology of them, but I think we are assuming that the island is a kind of closed population, so that the only sort of movement away from a study area would be via death. It is possible, so certainly, you know, the invasive species got on there because of people and boats, so it's not outside the realms of possibility that we could be losing some in that direction. But I think especially for the lesser night gecko, it's much more likely to be a bigger impact of the kind of predator population. So prior to that reintroduction, there was a lot of work to try and remove as many of the predators as possible, and to give them the best start by kind of having predator-proof fences and those sorts of things. But yeah, and certainly for some species, it would be an issue. And in fact, I think I've worked, I can't remember what species it was, but there was one that you did have to account for movement between the islands.
Yeah. Okay, thanks very much. I can't see any questions coming in. So I just want to thank Rachel for such an interesting talk. A really big thanks to Althea for inviting Rachel, and to the Caucus for Women in Statistics for organising the day and providing the opportunity to highlight the important contribution that women make in statistics and data science. The next presentation coming up in the next session is Women and Big Telescopes. So please do go to the IDWSDS website for details of the 36 talks that are being presented today. Thanks very much.